Continuing on with part 2 of Principles of Serological Testing. We've talked about the preparation of anti-human globulin serum, and now we're going to talk about the 3 types: polyspecific, monospecific anti-IgG, and monospecific anti-C3. First, we'll talk about polyspecific. Poly means more than one, and specific, specificity. This reagent consists of a pool of anti-human IgG, anti-C3b, and anti-C3d. These may be produced by hybridomas, rabbits, or a mixture. This may be used for routine compatibility testing, antibody identification, and the direct antiglobulin test. The most important function of this reagent is to detect IgG antibodies coating the cells. The importance of the presence of anti-complement in this AHG serum is very controversial for routine compatibility testing. Antibodies which are detectable only by their ability to bind complement are very, very rare, so it's not as useful for compatibility testing as many false positive reactions occur. So again, this is polyspecific. It contains anti-human IgG, C3b, and C3d. So when it's added to wash cells, it will detect IgG and or anti-C3 components. Monospecific anti-IgG is the most commonly used anti-human globulin serum. The major component is the antibody to human gamma chains. So it is an anti-IgG antibody. It contains no anti-complement and it's heavy chain specific. Every time you sit down and you use a blood bank reagent, you must briefly glance at the label. And when you're looking at your Coombs serum, you want to know, is it monospecific anti-IgG, monospecific anti-C3, or polyspecific? It is used as an alternative to polyspecific AHG in routine blood bank procedures. Now remember, polyspecific has both anti-IgG and anti-complement components. The advantage of utilizing anti-IgG is it eliminates false positives frequently obtained when using polyspecific AHG due to the presence of complement and nonspecific cold reacting antibodies. These can bind nonspecifically to red cells when stored at room temperature or especially at 4 degrees centigrade. So when we use polyspecific and we get a positive, we have to do additional testing. And most of the time, those are false positives. The monospecific anti-IgG will detect clinically significant IgG class alloantibodies only. Monospecific anti-complement contains anti-complement antibodies to detect complement coating the cells. It's used for classifying the coating proteins on red cells to determine if IgG, complement, or both are present. The primary importance is for classification of autoimmune hemolytic anemias and drug-induced hemolysis of red cells, because the clinical significance of if you just have IgG coating the cells or complement only coating the cells or both will determine paths for us to follow in doing more additional testing. For antiglobulin techniques, there are 2 applications of the antiglobulin test, and these drive students crazy. One is the direct antiglobulin test, DAT, or the indirect antiglobulin test, the IAT. The direct antiglobulin test is used to demonstrate or detect in vivo coating of red cells with globulin, particularly IgG and or C3. How you do the test is you take 1 drop of the patient's cell suspension, you wash it 3 times with saline to remove all contaminating proteins. Anti-human globulin serum is added directly to the washed cells. There's no incubation, There's no additional steps in the procedure. Take a drop, wash it 3 times, and add the AHG. You mix it well, centrifuge it for the specified period of time, and then observe for agglutination, which is indicative of IgG antibodies coating the cells. The antihuman globulin serum will bind to the IgG on the cells, forming lattices, which results in the observable reaction of agglutination. The uses, primary use is detecting maternal antibody coating fetal cells, which is an indication of hemolytic disease of the fetus and newborn. So one of the most common tests done on cord blood, the cord blood comes from the baby, is to take a drop, wash it 3 times, and determine if IgG is coating the baby's cells. If this is occurring, it could indicate increased destruction of the baby's red cells. It's also used to detect antibody-coated donor cells in a suspected transfusion reaction. So let's say that this patient is having a secondary immune reaction to these transfused cells, meaning they were exposed to the antigen before, they made an antibody. The antibody has gone below the level of detectability. We now have transfused that patient with antigen-positive cells, and they have a reaction. So what will occur is you will see that the donor cells will be agglutinated with the antihuman globulin serum because they're coated with patient antibody. It also is used to aid in the classification of autoimmune hemolytic anemias, which is a whole section on its own. So this picture nicely illustrates the direct antiglobulin test. So we take the patient's cells, we make a cell suspension, we take 1 drop of the cell suspension and wash it 3 times. In this illustration, we see that the patient's cells are already coated in vivo in the body with antibody. So we add 1 drop of the Coombs serum to the washed cells, and that will then cause lattice formation, which is a positive result. The indirect antiglobulin test, the IAT, detects in vitro sensitization with antibody, complement, or both. Patient serum, or sometimes special types of anti-serums, is added to red cells. They're incubated at 37 degrees, which is body temperature, for a specified period of time. Sometimes these tubes are read after incubation because some IgG antibodies may coat the red cells heavily enough after incubation to cause agglutination. How many times have I seen this occur in my lifetime? Very, very few. Probably I can count on the fingers of one hand. So usually these are not read after the 37 incubation. The red cells are washed free of contaminating protein. Again, the washing step is one of the most crucial steps in this procedure. And that is because the proteins, you have all types of immunoglobulins in your plasma, which could cause inactivation of the antihuman globulin serum. So after washing, you add it. If the cells have been coated with IgG during the incubation phase, the antibody in the AHG will form lattices, resulting in agglutination. So again, you cannot see sensitization. You cannot see antibodies attached to cells. All we can do is perform testing which will detect that this type of reaction has occurred. And the most common type of reaction we look for is agglutination. So this is an illustration of the IAT. So we have antibodies in serum, and we have red cells that are not coated with antibody. That's key. These are not coated with antibody. So what we do is we incubate the serum with these unknown cells, and during the incubation at 37, if the antigen is present to which this patient has an antibody, IgG will bind. We then wash thoroughly 3 times, and then we add the AHG to the test system. If antibody is coating the cells, we then get lattice formation, which results in agglutination. I need to burn this into your brain that the difference between the DAT and the IAT is, if you look at this picture, in the DAT, we're looking for cells that are already coated with antibody. We take a drop, we wash them 3 times, we add Coombs, and a positive reaction indicates in vivo sensitization of those cells in the body. In the indirect antiglobulin test, we're indirectly attaching antibodies to antigens. We have a patient or a serum that has antibodies. We have a cell with antigens. The cells are not coated with antibody. We put them together and incubate them at 37. During this time, antibodies will attach to antigens, and then the process is the same. Wash 3 times, add Coombs. So we actually attached antibodies to the cells by an incubation. They were not coated before. To determine that sensitization has occurred, we do the Coombs test. So DAT, we're directly detecting antibodies coating cells. The IAT, it's indirect because we are attaching antibodies to cells, and then going on to the Coombs test. There are other applications of the antiglobulin technique. The cross-match procedure may require the IAT, where you add patient serum to donor cells and incubate at 37, which is body temperature. You thoroughly wash and then add AHG serum. Agglutination of the cells indicates that the patient has an antibody to an antigen on the donor cells. So we would not give this unit of blood to the patient. It's called incompatible. When a patient has an antibody identified in their serum, it is critical to find donors which lack the antigen. Typing the donor may require the use of a typing sera reactive in the indirect antiglobulin test. So again, the Coombs test is a procedure that has many different applications. It's always crucially important that you know the limitations of your procedures, and this is especially true for the anti-human globulin test. Whether it's the DAT or the IAT, the following are reasons that you may obtain a false negative reaction, and you need to memorize all of these. The first and the most common is failure to wash the red cells adequately. As I stated before, your serum, your plasma is just chock full of antibodies to all kinds of things. We only want to detect antibodies coating the cells. So let's say that you're washing 3 times, but instead of 3 times you wash 2. Or that in between washes you did not resuspend the cell button adequately. This happens to students all the time. This will then result in a false negative because the anti-human globulin serum will react preferentially with unbound antibodies rather than those attached to the cell. And this is the most common cause of a false negative. If the testing is interrupted or delayed during the washing procedure, remember, antigen-antibody reactions are reversible. So if you have started washing and then you're interrupted due to a phone call or whatever, the antibodies that are attached to the cells can leach out into the saline. And then this will then, when you decant, you're decanting the antibodies that had been attached, and this can result in a false negative. Loss of activity in your AHG reagents. You never put droppers on the counter. If you remove a dropper from a vial, as soon as you're done with it, you put it back. 1 drop of a 1 to 1,000 dilution of human serum can inactivate an entire bottle of Coombs reagent. So if you have serum on your hands and you actually accidentally touch to the dropper to that serum, again, the bottle can be inactivated. failure to add AHG. And you'll notice that the Coombs serum that you're using is green. And this is because this was a real problem. I can tell because of my experience whether or not I've added Coombs reagent. I can tell because the volume is different. But as a newbie in the field, you may not. So the funny story is they had already approved blue for anti-A, the dye. They had already approved yellow for anti-B. So this really bright person said, why don't we put them together and then we'll have green and we can use this for our Coombs reagent. So this has been a wonderful addition because now when you spin down, after you've done the Coombs test and you spin it down and you're reading, if there's no green in the supernatant fluid, then you didn't add the Coombs reagent. So it's just saved you 30 minutes. Improper centrifugation. Under centrifugation, you're not allowing the antigens and antibodies to come in close proximity to each other, and so there will not be the lattice formation. Over centrifugation, the cells will stick so hard to the bottom of the tube that you're having to shake incredibly hard to dislodge it from the bottom. Because you're shaking so hard, if agglutination is there, you can have a false negative because you shook it too hard. The incorrect concentration of red cells in the test system, post-zone. So if you have too many red cells, even if sensitization has occurred, the antibodies are few and far between, so lattice formation cannot occur. Prozone reactions are when there's excess antibody, that all the antigen sites have been coated with antibody to where there's no way for your Coombs serum to come in and have a lattice formation. A negative DAT may not necessarily mean the absence of coding globulins. Polyspecific and anti-IgG will detect approximately 200 molecules of IgG per cell. So this is a light sprinkling. The presence of a weak antibody that less than 200 molecules of IgG are on a cell may go undetected. If a negative reaction is obtained, IgG-coated red cells, which are called check cells, are added to the test system. Agglutination ensures that the AHG serum is active. So what did I just say? The final step in either the IAT or the DAT is the addition of check cells. These are cells in a bottle that are coated with IgG. So let's say for every negative tube that you have, if it's 1+ or negative, you must add a drop of check cells. This will confirm that the AHG serum is active because when you add the check cells, you mix it very well, you centrifuge it, you must have agglutination. If you do not, you must start over. What is the most common reason for false negatives in the AHG test is insufficient washing. So the next time you do this, you want to be sure that you fill it 3/4 with saline, that you spin for 1 minute, that you decant thoroughly, and do not shake the tube when you're decanting. Because if you do that, you're going to rinse all your cells out, which is a whole nother story. But how you know that you've washed sufficiently is that your check cells check. Now we're going to talk about false negatives in the IAT. Remember, the IAT and the DAT are different in what they detect and how it's performed. Red cells in serum lose reactivity if improperly stored, which may result in a false negative in the indirect antiglobulin test. There are occasional examples of anti-JKA and anti-JKB that may be detected only in the presence of active complement. Active complement is found in fresh serum. And I've only seen this one time in my professional life. A colleague was working on a sample, and when she first did the antibody screen or the antibody ID, she was getting 3 to 4+ reactions in the tube testing. As time went on, because she needed to rule out some other stuff, she was doing additional testing. As time went on, the antibody went from a 3 to 4 plus to a 3 plus to 2 plus. Then she needed to do additional testing the next day. It was 2 plus to 1 plus because complement levels were going down. The strength of the reactions became weaker. The temperature and the incubation time affect attachment of antibody or complement to the red cells. So if it says, again, you incubate at 37 degrees, let's say you didn't check your heat block, which must be done and recorded every day. Let's say you didn't notice that instead of 37, it's now at 32. Well, this could affect the uptake of antibody onto the cell, resulting in a false negative. And if it says that you incubate 15 minutes, you incubate 15 minutes, not 14, not 12, not 10, but 15. Because if you don't, this could result in a false negative. In every area of the laboratory, procedures must be followed, and that includes the transfusion service. And my students would get really upset with me because I would be behind them, and I'm watching, you didn't do this or you didn't do that. Keep a ruler or a sharp edge, a sheet of paper, and put it under each step that you're working on. And then once you complete that step, move it to the next step. Because the most common problems or issues with students is because they didn't follow the procedure exactly. The optimal proportion of serum to cells should be achieved. If the reagent, the manufacturer says 2 drops of serum and 1 drop of cells, you cannot change that. Well, Terry said that if we have 3 drops, that would increase the amount of antibody. No, if the manufacturer says 2 drops, then you use 2 drops. Now let's discuss false positive results in the DAT and the IAT, which you must memorize. A false positive may be a result that the red cells may be agglutinated before they are washed. And yes, this has happened. But it's really odd because let's say that you're doing the IAT, and during incubation of the serum with the cells at 37, you spin it down and you have agglutination. Then you take it to the next step, which is the washing 3 times and adding AHG. Well, they were already agglutinated before you started washing, before the addition of AHG. But what I've noticed in the IAT is that usually the reaction will get stronger. It's 2 plus at 37, and then it's 3 or 4 plus at Coombs. Saline stored in glass bottles may contain colloidal silica particles that can leach from the container into the saline. They still mention this in textbooks. I don't know any transfusion service in the nation that stores their saline in glass bottles. We usually have a cube, and it's a plastic container enclosed in a cardboard carton. Improperly cleansed glassware. Again, we use disposable tubes, disposable pipettes. But let's say you're in a third world country where you don't have that luxury of throwing stuff away. Be sure that the glassware is thoroughly cleansed. Over-centrifugation can cause a false positive just because you've overspun it, and when you did that, perhaps the cells just decided to stick together. So again, if it says spin at 34 RPMs at 3,400 RPMs for 15 seconds, do not do 30. Do not exceed the time limit for centrifugation that is specified by the manufacturer. Improperly prepared AHG reagents, because the FDA is involved, I've never seen this as a problem. Every day before you use your reagents, you will perform quality control. And part of that quality control involves testing your Coombs reagent to make sure that there's no false positives and no false negatives. So if the Coombs reagent was improperly prepared, you would catch it during the QC. Let's talk about additional considerations for the direct antiglobulin test. Complement components, primarily C4, may non-specifically bind to the red cells from clotted blood samples kept at 4 degrees centigrade. So it's really important to allow your sample to come to room temperature, but even then, sometimes this reaction is irreversible. False positive DAT results may occur with blood collected into tubes containing silicone gel. So what is the purpose of the gel? Is to form a permanent barrier between the serum and the cells. How will you get to those cells? You're going to have to poke your pipette through the gel, and by doing that, you're then going to contaminate the cells with the gel. What is gel? It's sticky. So you can get false positives. Blood samples collected from IV fluid lines used to administer solutions. Again, these have different chemical makeups, which may cause the cells to be coated and react with our anti-human globulin serum. The more higher concern with this is that IV fluid contamination of serum will potentially dilute out any antibodies that we're looking for. Septicemia in a patient or bacterial contamination of stored specimens may cause a false positive DAT. One additional consideration for the indirect antiglobulin test is that red cells that are already coated with IgG cannot be tested with antisera that react only by the IAT, such as the weak D test or anti-Dupier present on transfused cells. There are procedures that are available for removing IgG from in vivo coated red blood cells. So I'll say that one more time. Let's say that you have a baby that's born, the mom has anti-D, you test them for the D antigen, and you get negative, then you have to do the weak D test. Well, if the baby has a positive direct antiglobulin test, you will get a false positive weak D test. How are you going to know it's a false positive? Because your D control, which must be run in conjunction with the weak D test, will also be positive. Remember, the weak D control must always be negative. What is the role of complement in the antiglobulin reactions? Complement binding antibodies attach complement to the cell surface when they react with red cell antigens. Some blood group antibodies bind complement to the red cell membrane. The clinical significance of complement binding antibodies is variable. Immune complexes present in plasma can activate complement components that they may absorb to the red cells in a non-specific manner. And this has been my finding, is that 99.9% of the time that I get a positive reaction with anti-complement, it's a false positive. Complement-coated red cells may or may not proceed to hemolysis. We've been taught that what is the role of complement? It results in cell cytolysis, destruction of the cells. But in our world, usually this does not occur. This completes part 2. Please be sure and read your textbook and study those objectives. Here are a few references. I would really like for you to spend time with the Indian Immunohematology Initiative. It has videos of the actual performance of blood bank testing with explanations.